sex bias. And that was the previous slide that we saw. And so welcome Melissa Wilson here from ASU. She's going to tell us all about genetic complexities of sex chromosome evolution. And the integrated theme that you'll find across the didactics and some of the elements of the presentations throughout the conference is on sex differences, and that was funded by the McDonald Foundation. Was very interested in investing in understanding sex differences, particularly in the context of cancer. Welcome.
All right, I'll completely ignore the timer up here. I'm not sure if anybody wants to deal with that, but I need a breath. Okay. Thanks. We have so much time together. I don't know if I ever told Kristen this. So first off, thank you, Kristen, for organizing and inviting me. I was a math major undergrad, and I went to a small school, Creighton in Omaha, Nebraska, and there were 8 of us math majors there. And then someone said at some point, you should go to this summer REU experience. So I applied to an REU in a math program and randomly got assigned to a group who did mathematical modeling. And I learned about Lotka-Volterra equations, and we modeled tumor dynamics as predator-prey with phosphorus uptake. And I thought it was the coolest thing. And it's what got me to apply, instead of only to pure math programs, to a bioinformatics program at Penn State, and why I have this whole trajectory that I do. And I know people talk about those REUs all the time, but it was super cool. And so I'm very excited to get to at least be a groupie so far of the math oncology group, coming back to modeling. I haven't done it for a while. I'm really a geneticist, genomicist now. So I'm at Arizona State University, and what I got into, so yeah, right, I applied to 2 programs, one in basic math at University of Nebraska and one in bioinformatics. It was brand new at Penn State, and down to like the April 15th, choose where you want to go. And I chose the bioinformatics program. I did a rotation with Katerina Makova, who studied sex chromosome evolution, and it was like all over for me. And so I'm going to nerd out about sex chromosomes for a while. Good luck to all of you. And I think I just used the mouse here. Oh. Excellent. Great. Okay, so we'll start out by talking about language. So people, when they're studying sex differences, say sex differences when they mean lots of different things. I mean genetics, because I grew up in a genetics lab, and I mean individuals who typically have ovaries and produce egg and have 2 X chromosomes, and individuals who typically have testes and produce sperm and have an X and a Y chromosome, sex chromosome complement. But I'm going to talk to you at the end about why this is so cool and wrong in so many cases. Some people might be talking about gametes. Some people might be talking about gonads or gonadal hormones, and some people might be talking about gender. Because I'm going to focus on that mouse the whole time if I didn't move it. Thanks. Sometimes the language gets confounded a lot. In clinical genetics, people don't assay sex chromosome complement, but they take reported gender, right? You can be male or female, or some additional categories may be allowed in checkboxes, typically not. And then we make assumptions about sex chromosome complement based on that. And again, my spoiler take home is that that's a super bad assumption to make, and I'll show you why. Okay, oh yeah, so evolution. So then I said, Kristen, can I talk about evolution? Like of sex chromosomes, because it's the coolest thing ever. And she was like, do whatever you want. And I was like, yes, this is why we work well together. Okay, so sex chromosomes are the most poorly named chromosome in our genome. We don't call any other chromosome by one function that it does. Sex chromosomes, there was actually a long history about what do we call them? What even are they? Should it be an X chromosome? Why is it called X? Why is it? It gets really fun and interesting. but there's only a handful of the genes involved in testes differentiation and ovary differentiation and spermatogenesis and oogenesis are actually even on the sex chromosomes. There is a regulatory element, sex determining region of the Y, that is important for the early differentiation of gonads, but that's it. There's over 1,000 genes on the X chromosome. Many of them do squat all for sex determination. And most importantly, sex chromosomes are not necessary for gonad differentiation at all. So this is, I'm going to show you one of these circle phylogenies, and here's a bunch of mammals. Mammals all have XXXY sex determination. All that means when we say XXXY, that means that it is the sperm producers that can produce gametes with one of two different kinds of sex chromosomes, here an X or a Y. Birds, just the opposite. They have ZZZW. So it's the egg producers that can produce an egg with either one of two types of sex chromosomes, either a Z or a W. Here, the W tends to degrade and be small, kind of like our Y chromosome tends to degrade and be small, but not always. Some birds, like paleognath birds, the large tinamous, emus, their W chromosome was like, I don't want to degrade, and it's quite large and homomorphic, almost exactly the same size as its Z. We can get into reptiles where they have XX, XY, ZZ, ZW, temperature dependent, environmental, and parthenogenesis. There's obligate parthenogenesis and also facultative parthenogenesis. So and also facultative parthenogenesis, meaning occasionally birds will also produce clonal offsprings of themselves. So the California condors here in Arizona were almost extinct. They did a breeding program with them, and they found that some of them, a mother, so who's ZW, because there's ZW sex determination. So a ZW will lay an egg that is a clonal reproduction of herself, but because then her egg is a clone, it's ZZ, so she will make a male clone of herself. So cool. Okay. Right. Okay. And there's obligate parthenogenic species. We have some on ASU's campus, whip-tailed lizards. We have a triploid species of lizards that the females, there's all females, they only produce eggs and will obligately produce only clones of themselves as a reptile. Right. And then we get into some other vertebrates, amphibians, fish. We just like to add on, right? So we have XXXY, ZZ, ZW, environmental, parthenogenesis, behavioral, where at one point in your life you can produce sperm, like the clownfish finding Nemo. Yeah. Okay. Mom dies. What happens? All the rest of the male clownfish fight. Whoever comes out on top stops making sperm, starts making egg. It makes that relationship look a lot different if you think about the movie. All of these eggs, other than the all of these species, other than the parthenogenic ones, have individuals who produce egg and sperm completely independent of whether they have sex chromosomes or not. We think that the evolution of sex chromosomes, there's a lot of hypotheses. We don't exactly know, right? It's an evolutionary question. Probably arose in response to environmental changes where if temperature is determining sex, for example, and your environment is not very stable, then genetics can be one solution to that, right? You can think about alternative sex ratios and whatnot. But the take home here is that determining sex can happen via sex chromosomes, can happen via XX and XY, but it doesn't have to. And even in humans, there's a ton of conditions where you have variations in gonad development that are independent of the sex chromosomes that you have. Oh, but here they are. Look, they're so pretty. And sometimes, so this is an X chromosome and a Y chromosome. Sometimes people will put the Y chromosome upside down to make it look like a Y, but it's not. This is how it's supposed to look. It's a acrocentric chromosome. Here's some mammals. For the newbies in the room, these were all the mammals that had any genome sequencing done when I did my dissertation. They were 2X coverage genomes. They were very bad, but they were very good at the time. And so we did some comparative genomics with them. And I just a context, right? Now we have hundreds of whole genomes, telomere to telomere. Like, it's just super exciting how far we've come. Oh, my favorite mammal, now I can't say that anymore. I used to say that, is the little one in the left corner, just turned 12. God. This will forever date me every time I give this talk. Okay, So a little bit more about evolution and then a little bit more about sex chromosomes. So yes, sex chromosomes are not required for sex determination, but how they evolved is really important and influences human health today. So the birds here, I'm going to orient you to just a smaller phylogenetic tree, diverged from all mammals about 300 million years ago. The monotremes diverged. So these are the egg-laying mammals. I'm showing platypus here, but there's also echidnas diverged from the rest of therian mammals about 177 million years ago. And then therian mammals, which is marsupials and us, diverged from each other about 150, 160 million years ago. So we can layer on top of that their sex chromosome evolution. So independently, birds evolved the ZW system. Monotremes did something cool and different. So I said all mammals have XY sex determination, but that again just means that it is individuals who produce sperm that can produce gametes with one of two different sex chromosomes. In monotremes, though, it's either 5 X's or 5 Y's. I wish I could say that's because platypus is weird. It's not. Echidna is actually even weirder because it lost one of the Y chromosomes. So they have 5 X's or 4 Y's. And that happened completely independently of the shared XY system in the therian mammals. Marsupials have this shared XY system between marsupials and us, which are eutherians. And then eutherians, we, in the common ancestor of humans, dogs, mouse, rat, whales, we added a 50 million base pair region to our sex chromosomes. It's shared across all eutherian mammals. There's another layer I usually put on top of this, which is placentation. I took it out for this group, but I just want to mention that the evolution of the placenta also mirrors this, where monotremes, birds have these hard-shelled eggs, monotremes have these kind of leathery eggs. Marsupials and eutherians actually both form a placenta. So it's not, you should not call eutherians placental mammals because you're excluding all the marsupials that actually do form a placenta, but it only lasts for a few days. And then only in the animals that have this X added region, Y added region, do we also have a fully formed integrated placenta that lasts for, depending on the species, days to years if you're an elephant. But I think that the evolution of our sex chromosomes may have allowed that to happen. I can read more about it here, or I can chit chat with you about it. Okay, so our sex chromosomes about 160 million years ago, maybe 200 million years. We're evolutionary biologists, so like, what's 40 million years? It's a give or take. Our X chromosome has about 155 million sites and 1,100 genes on it. Our Y chromosome has about 60 million sites and only 27 unique genes. Here's what I like to point out. The Y chromosome is not the smallest in the genome. It's actually smaller chromosomes. The Y is the most gene poor, but it's not the smallest in terms of overall nucleotide base pairs. And that's important because it encodes also a lot of regulatory elements, a lot of multi-copy genes. Some of these are parts of a gene family where individuals who have testes and make sperm need at least 40 copies of one of those genes to be fertile. So there's lots of genes here. They do lots of interesting things. There's been some cool work in Drosophila showing that if you take the same autosomal background and put a different Y chromosome into multiple different Drosophila lineages, you get wild differences in gene expression on the autosomes. As of yet, we haven't done this in humans, but it might be something we could do in the future. We can't. I'm sorry. I don't have a great sarcasm filter. I just use the same voice. So we're not going to take humans and do genetic crosses on them. Okay. This is my favorite plot in the history of plots because on the x-axis, you'll notice the X chromosome. And on the y-axis, the sequence of the Y chromosome. And this is the sequence similarity between them. And you can see that there's not a lot of little dots in there. This is called a dot plot. It's something that we used to do in the olden times when we actually looked at whole chromosomes of individual species. And now we look at hundreds of thousands that we don't visualize them as much anymore. But the big take home is that there's regions like this X added region that I talked to you about that only eutherians have, shares a lot of sequence similarity between the X and Y because it's more young evolutionarily. And you can see in the bottom left-hand corner is that pink little pseudoautosomal region, which is 100% identical between the X and the Y. Oh yeah, there it is. And then there's a second pseudoautosomal region on the other side. And there's also something that's uniquely human called an X transposed region that was duplicated from our X to the Y. And so there's these three regions at least that share between 97 to 100% sequence similarity between the X and the Y chromosome. So why does that matter? So I guess this is, I mean, it's all teaching, but a little bit more about the technical, why does it matter and why should you care if you do genomics, unless you listen to our first talk, in which case you don't have to care about genetics. So you should all just space out for the rest of this talk. It matters because of misalignment. So here's a human karyotype. Typically, individuals have 2 copies of every autosome, one inherited from each parent, not always, right? You can have 3 copies. And typically individuals will have 2 X's or an X and a Y. When we're doing genome alignment, we don't show a representative of every chromosome. Instead, we just use one haploid representative for every chromosome in the genome, except X and Y, which we keep in there completely. And that's not right. It's not right and wrong. Sorry, I'm such a Nebraskan. I'm like, rather than being like, that's wrong, I'm like, that's not right. That's not quite right. Okay, yeah, it's wrong. Okay, and it's wrong because it means you get misalignment. So for let's start with the XY individual, right? So their chromosome 8 is our representative autosome. They have the maternal, paternal chromosomes, chop them into short reads, align them to our 1 copy of chromosome 8. Cool, that works all right. X and Y, chop them into short reads, sequence a bunch of cells because we're doing bulk sequencing here. And then you might get a little bit of misalignment between the reads, between the X and the Y in those 100% identical regions, the tips, the pseudoautosomal regions. We know that happens, but most people don't care. So what about an XX individual? So chromosome 8, again, align it to your standard single haplotype representative. But our 2 X chromosomes, what's going to happen is that some of the reads from the X are going to align to the Y across the entire Y because of that sequence similarity that I just talked to you about. This is a problem across all major human genome sequencing projects. And we found this out because, gosh, like 5 years ago now, I had a student that I said, go pull down the Y chromosome BAMs. So pull down the alignments for the Y chromosome from this major redacted name sequencing project and do PSMC. We wanted to look at evolutionary history of the Y chromosome, right? And she came back and it looked super weird, right? So evolutionary history of the Y chromosome for about half the samples looked like nothing. Like there were some variants there a little bit, but like everybody was coalescing really soon and like, okay, right? Like that we're not here talking about PopGen, but the reason it was weird is because she pulled down the BAMs and everybody, the XX and the XY individuals all had reads on the Y chromosome. So it's wrong for 2 reasons. 1, it makes people think there's Y-linked reads in an XY individual. And 2, all those reads belong on the X chromosome. So now you're not aligning reads to the X where they belong. Oh yeah, so what do? Let's get rid of it. So we came up with a software approach, XY Align, where you could infer the sex chromosome complement of the sample that you're working on. You can do a bunch of quality control that you want and then realign with appropriate sex chromosome mask. So what that means is if there's evidence of a Y chromosome, so here we don't care how many X's, right? Just like you don't care how many chromosome 8, how many chromosome 20, you don't care, you just care, do you have it or not, right? So if you have a Y, then mask out the regions that are 100% identical. If you don't have a Y, hard mask the whole Y chromosome. I was wrong, you can't actually remove it completely because then your downstream software won't work, and we figured that out really quickly. So we hard mask it, so it's still there, but we don't allow anything to align to it. Okay, and this is a table, and I try not to put too many tables in talks because nobody wants to see tables, but this is in a single sample. How many single nucleotide polymorphisms, SNPs, mutations do we call in a sample if you do the sex chromosome complement aware approach versus a standard? And it's almost 10,000 more. Okay, in a genome where you have millions of SNPs, that's not a lot, right? If we're looking at less than 5% of the genome, but this will aggregate across multiple individuals as you think about missing SNPs, missing calling variation across sex chromosomes over and over and over and over again. So we're missing a lot of variation there that we could recapture easily, right? Okay. So that was for DNA. We also showed that this works for RNA. And so if you're doing RNA differential expression, maybe unsurprisingly, so on the left is what you call the default approach, and there's our table again, and on the right is the genes that you call as differentially expressed using a sex chromosome complement aware approach. We get one more Y-linked gene, and we get one more X-linked gene, and we get a few more Y-linked genes, and you'll say, like, isn't it kind of obvious that if you're comparing between XX and XY samples, you should have differential expression on the Y chromosome? Like, yeah, but we're not calling it using a standard approach because of the mismapping between them. And so if you're just running these pipelines over and over again, you're missing sex differences in gene expression on these chromosomes in genes that are not involved in sex determination, that are going to be involved in multiple other processes. Oh, yeah, and this especially matters for genes that have a functional copy on the X and a Y. So, right, let's go down the rabbit hole a little more. So I talked to you about why sex chromosomes are cool and they evolved and they have the coolest evolution and they change across species. And then there's these genes that still are retained for 180 million years. They have a functional copy on X, they have a functional copy on Y, and they have survived. Everything else has been lost, all but 27 genes. And even half of those are recruited. So like all but about 17 genes. And out of over 1,100 genes, these like 12 to 19 genes have been retained for 180 million years. They got to do something important, right? Okay, well, that's a fallacy in evolution, but we've proven that they do important things. So trust me. Okay, these genes. So what could happen in our standard approach on top and our sex chromosome complement informed on the bottom, the green on the left is in our XX samples and the orange on the right is our XY samples. And on the top is if you only count the X chromosome expression, and on the Y is, or on the bottom is if you count the expression from X and Y. And so what could happen is squat all, nothing. You do this approach and nothing changes. So cool, Melissa, why did you tell me this? Oh, because sometimes it makes a big difference where you go from having no sex differences in gene expression, so this gene PCBH11X, to showing a significant male bias in gene expression. You could go, again, from nothing. You could be differentially expressed in both, or you could be not differentially expressed in both conditions. Or you could go from being differentially expressed to actually not showing any difference between the sexes. So ZFX, ZFY is highly conserved on the X and the Y in all mammals that we've looked at. This is a super highly conserved gene. We think that the function of the Y-link copy is important. And if you're doing your standard approach, you would think that it's differentially expressed between XX and XY individuals, but it turns out it's not in the tissues we're looking at. Or you could even change direction. You could be showing a bias towards higher expression in XX individuals and switch to a higher expression in XY individuals. So just to say that for a small number of genes, and genes, some of these have been shown to be really important in some cancers like multiple myeloma. Understanding how they're being expressed accurately in XX and XY individuals could completely change your interpretation of the data. Oh yeah, okay, so here's the human sex chromosomes, and why should we care? Why should we just not take those check boxes? One, that's lazy, and we're not lazy. Nobody here is lazy. Two is they actually vary quite a lot. So I didn't talk about the other piece of sex chromosome evolution at all, but that other piece is X inactivation. So individuals with two X chromosomes have 1,100 copies of both of those genes. Individuals with a single X and a Y only express copies from one. There's no other chromosome in our genome where you can survive with a single copy of that chromosome. You can't have a single copy of chromosome 1. You can't have a single copy of chromosome 2. It's not compatible with life. Making a big assumption that many people in this room only have a single X chromosome and they appear to be doing just fine. That's because of a process called dosage compensation, where X individuals will inactivate one of their two X chromosomes. It's actually a super cool interplay. Again, later we could talk about how that happened. But it means that then you actually have very similar dosage between the active X chromosome in X individuals and the single X chromosome in XY. And then you have a reduced expression. It's actually not completely silenced. It's reduced. And individual genes escape and it gets really fun, but it's reduced. And that means that you can actually have a lot more variation in sex chromosome karyotype than you can with any other chromosome in the genome. So individuals with two X's and a Y who develop testis it's a condition called Klinefelter, occurs approximately 1 in 500 live assigned male at birth. Individuals with a single X chromosome develop ovaries and female characteristics is approximately 1 in 2,500 live assigned female at birth. These cases, actually, individuals with a single X chromosome, now that we've started looking more with genetics and genomics, almost everyone here is actually mosaic. They all have either a piece of an X or a piece of a Y. But recombination is a wild beast and messes up sometimes. On top of that, individuals with an X and two Ys, 1 in 1,000, live assigned male at birth. 3 X's, similar, 1 in 1,000, right? You're seeing a trend here? Individuals with 2 X's and 2 Ys, 1 in 18,000, and then individuals who have an XY sex chromosome complement, but a deficient SRY, so develop female reproductive tract, is 1 in 20,000. So if we start to think about our cohorts getting larger and larger and larger, we certainly include many individuals with sex chromosome karyotypes that are not the typical expectation. But it gets cooler. Y chromosomes are lost over life, and they're lost in cancer. So as individuals with a Y chromosome age, their cells will lose Y chromosomes over time. And the proportion of blood cells in which an individual has lost the Y chromosome is actually predictive for many blood-borne cancers and also dementias. I often get a question, what about the other tissues? Well, we don't usually biopsy people's livers or lungs or hearts or others just for funsies. And so I don't know. But I know that in the blood that we can characteristically assay, the Y chromosome is lost over time. And in many cancers where it has been assayed, we have a lot of data, we haven't looked at it, but in many cancers, the Y chromosome is also lost. Yeah, yes, please do.
Right. So I remember hearing that in lymphocytes, people can see like accumulation of micronuclei with age. So this is like, a crude microscopic measure. You can see accumulation of micronuclei with age. It's very elevated in smokers and elevated in people who are deficient in vitamin B12 and folate needed for DNA repair. Do you know if that's correlated with this sort of thing? Like, you've got this one sort of simple pathological, way of observing accumulating genetic damage in blood cells, which probably goes hand in hand with this insofar as you describe this correlation with like higher risk of blood cancers.
So I think that's a great question. To my knowledge, largely with this with the increased loss of Y chromosome with age, it's just been a straight up observation just in large biobank cohorts. So nobody's correlated that with anything else, but I think that's a great idea, right? If you have cells that are wonky, are they wonky in many, many ways?
Yeah.
Perfect, right? That was the technical term there. So I want to, I guess the take home is that sex differences are multidimensional, and my lab focuses on this very small genetic component of it, where we think about sex chromosome complement in individuals as they vary in a population and as it varies with time and across the kind of samples that you're collecting. But on top of that, we layer then variation in gonadal hormones. We layer on top of that gonad development, right? So you can see even in a 8-cell stage individual, sex differences in gene expression prior to gonad differentiation, prior to any hormones, but then you have hormones coming in, playing a role in gonad differentiation in pre-puberty individuals, throughout puberty, through menopause, where you have massive shifts in disease risk, interplaying with the genetics, right? So it all gets complicated and fun very quickly, but hopefully just to say that the genetic component of it is, I think, going to be important. With that, right, so Kristen pulled me into this, thinking about this, sex differences relevant to cancer, right? I didn't do cancer at all after my REU. And then cancer has this way of pulling everyone back into it. And I think that's really valuable because it lets people get some experience in a different field and then come back and apply that here. So we've started now to think about distinct molecular etiologies in cancer based on the sex chromosome karyotype of the sample. So with that, thanks for our funding and ASU for giving me a job and keeping me on. And thank you all for listening. Questions, fights, pushback.
So what do you think the, of course, I'll have a ton of questions.
First of all, thank you. But I was wondering, so I know there's been, I think, some studies that if you start giving someone, say, hormone supplements, they'll stop producing it. And even after you take those supplements away, they still won't produce the hormone anymore. And I was wondering, do you think it's possible that that sort of speeding up maybe the degradation of these chromosomes and partly causing that?
Do I think that hormone supplements could affect chromosome retention in a cell over time? I only know about more mutagenic effects on an individual level and not on cell segregation. So other people might know, and it might depend on the kind of hormone and whether like estrogens and androgens act pretty differently. So I don't know. I think it's a good idea though.
Thank you.
Yeah.
Hi, I have a quick question about the relationship between aging and cancer and loss of Y chromosome, basically. Just I was wondering if you had a chance to look at the pediatric cancers and loss of, for example, Y chromosome and sort of, not.
No,
aging.
that's a really great idea. So we just have a proposal right now with Kids First data and collaborators at CHOP at the Children's Hospital of Philadelphia to look at pediatric cancers because similar to adult cancers, largely showing a male bias in incidence and worse prognosis, pediatric cancers also do. And to my knowledge, nobody's looked at the sex chromosome complement in those individuals where you would expect there to be more of an effect, right? So yes, you should review our grant. It's going in this summer and fund it, please. Yeah, I think it's a great idea. And if anybody has pediatric cancer data, please go look at it, right? All of my ideas are yours. Please take them and run with them if you think are good. Thanks.
Thanks, Melissa. That's a great talk as usual for you. Could you tell us a little bit about the paper you just flashed up there with Kim Buteau about sex and sex and cancer?
Yeah. Oh, yeah, sure. I didn't put much in it here, but so there's a struggle. I struggle with how to appropriately incorporate sex as a biological variable into analyses of sex differences, because inherently by segregating your data into individual samples with XXXY, then you lose power. And so part of what we've done, and we talked to Barbara Stranger and other people who've done work here, is we do segregate out our samples to look. So we looked at we looked at tumor adjacent differences and tumor normal, which was whole blood in that sample, to identify tumor specific mutations and tumor specific gene expression. And then we segregated the samples into males and females, and then used a relaxed p-value threshold in the female samples, because liver cancer, at least in this cohort, has about a 4-to-1 male to female incidence. And so in the cohort, you just don't have as many females. It's a challenge, right? And I think, sidebar, I think collecting just more females isn't going to be representative, because you're actually going to have more heterogeneity in the sex that has a higher incidence. So all that to say that about a third of the sex differences in DNA mutations and in gene expression were shared between males and females, and about a third of them were unique to females and a third were unique to males in terms of the individual genes that were differentially expressed. In females, we saw mutations involved in recombination, and I'm forgetting exactly the mutations we identified in the male samples there. This cohort that we looked at was from the Cancer Genome Atlas, and we didn't have viral mediated in it. So we just kind of lumped everyone together. And so we're redoing this, looking at viral mediated liver cancer also to see if there's some differences.
So nice to listen to you talk again, Melissa. Just a comment about the sex chromosomes in pediatric cancers.
Yeah.
So in that the X chromosome has a number of tumor suppressive functions. In medulloblastoma, where it's best studied, there's a subtype in which there's very frequent loss of an X chromosome, highly malignant tumor, and you see loss of X because presumably that's part of the oncogenic mechanism. The leukemias in kids, like those in adults, have loss of Y chromosomes. Just wanted to comment on that. And I'd love to hear your thoughts about the fact that the X chromosome does have a number of tumor suppressive functions, particularly the things that the Haups have described with regard to interacting with the P53 pathway, innate immunity functions that escape X inactivation, and then the difference between TSPY and TSPX, whereas TSPX is a tumor suppressor and TXPY, which is almost identical, is an oncogene.
Yeah.
So with regard to chromosome dynamics and cancer, I think there's a whole other level by which these chromosomes, they obviously sex determine, and they obviously influence sexual differentiation, and sexual differentiation influences cancer biology. But intrinsically, they also have tumor suppressive versus oncogenic function.
Yes, sorry, was that my shut? Okay, it's not my shut up time. Okay. I saw people going over time a little bit today, so I'm going to just assume we're going 45 minutes over. You all can eat your lunch in here. Yes, because X inactivation and dosage compensation is the wildest evolutionary process that has happened. And we have been so reductionist in our thinking of it because, as Josh mentioned, there are at least 40 to 50 immune-related genes and tumor suppressor genes on the X chromosome that routinely are not subject to X inactivation. They routinely escape. But there's also some that escape only in a cell type specific way. And there's individual genes that are expressed in the B cells and T cells only. And we didn't know that because we were just taking one tissue, assume the tissue looks the same across all of them. And I think that getting at the role of sex chromosomes in cancer is going to take a lot of careful project design because there's the immune related component of it. TSPX, TSPY is also tumor suppressor and oncogene is wild. Also, TSPX is related to viral degradation in the liver, and TSPY lost that function. So if we think about viral mediated liver cancer, you can have the same gene. One is a tumor suppressor gene on the X chromosome and degrades viruses on the X chromosome. Individuals with two X chromosomes have two copies of it. It escapes inactivation. And in the Y chromosome, it gets a double hit. It's oncogene, and it doesn't have the capacity of two genes, two copies of a gene degrading viruses in the liver. And so you could, not that that's the one be all end all gene, but I think that it's more than just understanding whether you have a full Y chromosome or not. Do you have partial Y? Which genes are escaping on the X? Are they tissue specific? Are they escaping in your cancer of interest? How are those immune related genes related to response to immunotherapy? How are they relating to response to any sort of treatment? Because inflammatory responses can be just massively over responding, right? And so my brain explodes a little bit thinking about it, but in the good kind of, because I feel like adding the genetic component of sex chromosome variation across individuals is going to help like it's going to help us make a difference in people's lives, right? And that's like an overwhelming and positive thing and frustrating thing that it's been ignored for so long. Sorry, now I don't know if that was a great answer to your question, Josh, but yes, I think that the immune related function of genes there and the tumor suppressor function is going to be really important.